Before I start talking about OverCITE-seq, a bit of a historical background of how these technologies were developed at the Genome Center, which is my host institution. So we started, I think, like 3 or 4 years ago, with developing the first iteration of this single-cell genomics called CITE-seq. And the advantage here was that you could capture the single-cell RNA, so the whole cell transcriptome, but also capture cell surface proteome of these cells using antibodies that are barcoded with DNA oligos, so that you could quantify at the protein level how the cell's state changes as an additional layer of information on top of the single-cell RNA-seq. So that was the first sort of technology. Then 2 years later, we had another expansion of that technology called ECCITE-seq, where we added CRISPR perturbation to the modalities that can be captured in the single-cell experiment, and then you can do functional genomics. So you can take out certain genes and then look at how that affects the transcriptomes and cell surface epitopes in those cells. And then the third generation of these modifications was OverCITE-seq, where instead of capturing the CRISPR guide RNA in the knockout screens, we capture the overexpressed genes. So as you remember in the study that we're describing here, we overexpress a range of different genes in T cells and look at how they affect T cell states. And with OverCITE-seq, we can assign to each single cell, coming from different T cell subsets, what genes are overexpressed there and what effect globally on cell transcriptomes and surface epitopes they have.